through the intracellular receptors. So when we talk about proteins, when you talk about any protein hormones or protein factors, they all work at a cell membrane level. So they stimulate the growth or proliferation of the cell by binding to a specific cell surface receptor, right? That's important in the cell, and provide cellular communication. They are mitogens, they enhance cell growth, and it's very central to the signaling pathway. So majority of the signaling pathways have received their response, they respond to the growth factor action. In the growth factor is the essential thing. It has to bind to the receptor, transduce all the function, and then ultimately the cell is going to have a response. There are several growth factors. The liver has a hepatocytic growth factors. Majority of them come from the brain also. You have the bone related factors. You have the growth factors that are coming from the pituitary or hypothalamus. So hypothalamus, pituitary are the main organs providing you growth factors, but there are other organs that also can provide generate growth factors. Okay, there are two kinds of growth factors basically. The ones that are competence growth factor, they stimulate cells to enter G1 phase from the cell cycle. So you need growth factor for entering from G0 to G1 and then maintain into the G1, right? So the ones that are required for cells to enter into G1 are called competence factors. The factors that are required to maintain G1 phase or during the progression of G1 are called the progression factors. They are necessary to ensure that cell will enter S phase. And as we said, that growth factors are important only during the G phase, and once it reaches the restriction point, the growth factor function is over. So now you have two different divisions, ones that occur before pre-G1 and one that occurs late G1. So this is the same, saying the same thing again, that competence factors allow the cells to enter into G1 from G0, and progressions will go through the later on that will allow the cells to come. It's the way I've shown here is a quiescent or resting stage, and then you have progression factors, and then you have the no growth factor requirement. Now I have to take a step back, because what happens is the entire growth factor function, epidermal growth factor is one of the main ones, or TGF alpha, they all have some connection with the oncogenes, and so we have to kind of review our approach to learning of viruses. Just a couple of slides, okay? So what happens is that the viruses would affect the proto-oncogenes to make oncogenes. Either it can be mutation, or here we are talking about just the viruses right here. So what happens is that the oncogenes, then we know, that affect the growth factors will change the signaling cascade, and that will make normal cells have normal cell proliferation. That will make a transformed cell. Transformed cell proliferation will make cancer. So what these viruses are, and that's what I really want to kind of spend some time on, is and here we summarize saying that they are activated by a variety of mechanisms that encode growth factors and receptor kinases and other enzymes that participate in mitogenic signaling. But then if you look at it, the oncogenic viruses are, you keep your mind clear that your viruses that we know as viruses are different than what we are going to talk about cancer. So in other words, there are very few viruses, hardly any really, that you can put that in the human and it's going to cause cancer, right? Except that you have the HPV for cervical cancer or something like that. But there's a long time ago, there's a guy named Dan Moore in Washington State University, and there was a science paper, I think it was science, I may be wrong, But there are a couple of papers from his lab saying that the virus can induce or can cause breast cancer, and he had isolated viruses from milk, and somehow that research has, just did not go forward too much for a variety of reasons which I don't know. But we didn't see too many papers after that. So viruses causing cancer is not as frequent, but viruses causing influenza, other diseases is quite common, right? And then you have retroviruses and you have DNA viruses, and here we are talking about retroviruses. So you have the viruses that transform cells. If a virus integrates into a host cell genome, it will change its property and cell becomes transformed. Now what happens is, as I said, that there are two kinds of viruses. You have DNA virus and you have a retrovirus, which is a RNA virus, and this is just the definition of oncogenes, that the protogenes altering its function are called the oncogenes. Now this is important. What happens is, whole concept came from the Rous sarcoma virus. There's a huge background on Rous sarcoma virus, because that was causing tumors in the animals. So what they have is, they have a sequence called SRC. Now this SRC is a envelope, like that's an extra gene which is required in a virus by itself. It's a structural protein thing. But this RSV gene, which is SARC by itself, has not functioned. It's not required for viral replication. But many times this extra gene is found in the Rous sarcoma sequence. Okay, so remember the word is SARC. SARC was also found in many human tumors. So that's why the research on RSV or Rous sarcoma virus was really intensified, and I'll go into detail. It will become very clear why it's important. The SARC protein is a self-phosphorylating protein. That's the only protein in the body that self-phosphorylates. Other proteins require, you require ATP or some sort of a phosphorylating influence, whereas SARC is a self-phosphorylating protein. It is divided into 3 domains, and then you have a different action for different domains. The SH1 is a catalytic domain, whereas they are involved in the substrate regulation of the catalytic activity. Okay. Now, if you look at that, just the names, so the Rous sarcoma virus, you have the S is called VSARC if it's in the virus, and they're all in the VCs, the V meaning the virus, right? And they're all found in the tumors, and then that is thrown out. So the SARC, MYC, all those are not, are not really viral anymore once they are in the tumor. If you look at the growth factor signaling in a bird eye view, then I showed this slide for 1 or 2 reasons. One is, as I was telling, that all growth factors across the board, the function is the bottom line. Either you are going to have a apoptosis, you're going to have migration, you're going to have growth, you're going to have cell adhesion, or you're going to have a differentiation. So this picture is also from Weinberg book. But what happens is that you look at the 3 main parameters. You have growth factors like TGF alpha, EGF. The difference between EGF and TGF alpha is they are very similar in nature, except there are some sequences in TGF alpha. So EGF by itself does not cause tumors, TGF alpha can. So TGF alpha is carcinogenic, whereas EGF is not. Then you have all these different growth factors, neural growth factors are there, several of them. They all bind to their own selective receptors. So these are this cartoon shows these as receptors, and then they are internalized, and turn on the signaling cascades a variety of different kinds, which in terms of going through either the the PKC pathway, or it goes through the MAP kinases, or you go through the junk pathway, and they all finally end up having some sort of a transcription differences, which will make the final product differences. So if you look at the sideline here, so we have the these are all the signals, or called ligands. These ligands are the growth factors. You have the receptors, they bind to them. They have the adapter proteins, which varies, and they have to sort of be regulated by binding itself, and will go just like cell cycle. We know G1S and G2, but then when you go into it, it's just, it's quite complex. The same way here, the adapter proteins and enzyme functioning is complex. Then you have a signaling cascade occurring, and then you have the transcription factors in the genome level inside the nuclei, which in turn alters the genomic profiling, which causes either apoptosis, cell division, growth, adhesion, or cell differentiation. Now, when you look at it, the one thing we have to keep in mind that once we identify a signaling process for any molecule, then you know that if there are inhibitors available, something that you can stop, then it will stop that process. So for example, if you have something required, just an example, this is wrong, I mean this is not, I'm just taking it as a hypothetical, that if you find an inhibitor that would inhibit conversion of PI3K to AKT, right, then whatever the AKT function is later on, going through the FOS and increasing cell proliferation, will be blocked. So if the cells, if you have a cancer cell that are growing so effectively and it's following this process, then you can find a cell block, or you can do something that blocks that step of processing in a signaling, and that should be your aim. So in other words, if you want to do a study on say compound X, and you identified that it works through whatever pathway, beta catenin or AKT, PKB, whatever pathway that it works through, you have identified a pathway, you go through the literature, find an inhibitor that inhibits a single process during that cascading event, and see if that works to prevent or suppress the growth of your cells in the, and then you can select that as your starting point for writing your proposals or writing your grants, and you can come up with new ideas and submit it, and then you can go forward with a big time research and make that as your aim. And that's basically all of us did in the past or are doing. That's exactly what you do. Sometimes you have you don't have to identify cascade by yourself. You know what it acts on. So the signaling cascade is already known in the literature. You pick a step, you confirm a step that okay, I have this expression occurring and the next expression occurring, and there is overexpression when I put my compound in, or under expression suppression. Then you reverse it and see that okay, if I find an inhibitor that's available in the literature, or that you know of your own that you are working on, you see if it blocks it and your cell inhibition is occurring at the end. Well, that's all you need to know. Once you know that, then you are home free. Then that's a starting point for your next 10 years. So that's how it works. Okay. So let's look at the organization of the EGF receptor. We won't do too much on this, but it has an ectodomain, like where the ligand is binding. Now when we showed that cartoon, you look that as upside down. Okay, then you will get a good relation. See, like for example, here we did it like it went from top to bottom. Here you see then and turn it around, turn this gene around. So your EGF receptor gene, we have ectodomain where the ligand is going to bind, then it's going to come down, then your transmembrane is going to cross the membrane, get inside the cell into the cytoplasm. Once you are in the cytoplasm, then you have the C-terminal end, and that C-terminal end is going to have function. The most important part to remember is that in EGF receptor structure, you have a Src homology. So there is an area in there which is identical to Src. Now, as we said in the previous slide, that Src is carcinogenic. That is responsible for causing cancers, right? So if you have in EGF receptor your Src, that means it's prone to causing cancer. That's the thing you have to remember. So the Src, as I said, is a self-phosphorylating protein, and it's a phosphoprotein, means it self-phosphorylates. That's the only phosphoprotein that can be phosphorylated, or there is nothing in the body that does that. And we talked about tyrosine and serine phosphorylation. Tyrosine phosphorylation is for mitogenic signals, and serine phosphorylation is for the CDKs, and also TGF. TGF beta is the only growth factor that uses serine phosphorylation and not tyrosine phosphorylation. All other growth factors use tyrosine phosphorylation. Okay. Now the ERB is very important, because what happens is that you have a I'll talk about it. We have the in breast cancer we know about HER2/neu that's coming up, and the ERB originally was present in the animals, in the rats, and then it was transcribed into the or identified into the human. So if you look at the comparison between the EGF receptor and ERB, you have a lot of similarities in the in its genomic in the gene structuring. So all that's trying to say that EGF receptor, ERB, and HER2/neu, they all have very similar structural organizations. And here it shows the sequencing. We don't want to worry about that. You have a sequencing similarities in them. So the EGF receptor with kinase activities, or if you look at the TGF alpha, I say the TGF alpha is the one that's going to cause tumors. You have a EGF receptor family. You have its cutoff is a FGF receptor or PDGF receptor. And if you look at these ones in the parentheses, those are the oncogenes that it really is trying to work on. For example, these are all the oncogenic compounds, right? So many growth factors, as I said, like which are responsible or are considered to be responsible for having the carcinogenic activity, they all have their homologies with respected viral component of oncogenes. Okay. So these are some of the growth factor comparisons that we have shown. They all have the similar layout. This is the tyrosine kinase domain, which is important, and that's how it's going to go in. The phosphorylation, some of them like insulin, like growth factor has, as a duplex, has two gene thing. This NGF, or they all have a different layout, but they all have similar cascade of arrangement of all of the structures. So what happens is that some of the growth factors, they function differently. Normally what happens is you have a growth factor that binds to the growth factor is going to bind to the receptor here, and your tyrosine kinase, and is going to cascade all the function. Some of them is going to have the ligand dependent firing. It means you have the phosphorylations occurring with more and more growth factor or growth factor like component coming in. You have the phosphorylations occurring. If you have a mutation occurring in that, then what happens is that it does not require the ligand. So now you have a receptor that's autophosphorylating, and one of the reasons you want the cell to do is that you have a growth factor receptor, growth factor binds to it, you have a cascade occurring by phosphorylation, having the tyrosine kinase activity. Now sometimes what happens is that there is a mutation and the growth factor receptor will auto fire. They will not require the growth factor, and that's the abnormality that can cause cancer. So I'll show you the slide what you do with it, and this is just to show the immunofluorescence. Now what happens is, this is just a technique point of view. Don't look into too much into the picture, but a lot of confocal microscopy is used these days. And the confocal microscopy, what they do is they look at the merging. So they have like a EGF, and they have EGF receptor. They stain two different ones, two different sections. They try to merge, see if they are merging, meaning they are binding to that particular protein. Now for known proteins and receptors we know, but for the unknowns it's a really good technique. There are a lot of problems with confocal microscopy and the merging. Is many times you see backgrounds, and I'll talk about it if I have time, because I had a paper on that, saying that what happens is that if I had a protein and I had a receptor, and we merged it using confocal microscopy, and it looked just like that. So that you have now yellow, you have green, you have red, you merge it, you look yellow. Then you do the immunoprecipitation studies using biochemical techniques, and it does not bind. So what that means is that what you always see in immuno using the merging process is not always correct. And what I did is I got a paper out of it. Of course, I just wrote a paper saying that you cannot believe this all the time, and then proved it. So as I was saying before, you always should depend on more than one technique if you can. If you have facilities and resources, try to do it for more than once, because there are many things that you do in science, it looks great, and then it just has so many false negatives that you have to pay for later on, because when you go advanced, it probably won't work, and then you'll be frustrated that, well, I should have kept more attention in the beginning. Okay, and this is, I'm going to go show just the another, all receptors work differently. So as I told you, TGF beta is the only growth factor, or its receptor, TGF beta receptor, that uses serine threonine kinase domain, and they work separately. And all they do is, for example, if you look at EGF, the two units phosphorylate by itself. In TGF, they phosphorylate the opposite one. So the tyrosine kinase domain is here, and it phosphorylates the other one. So there are some differences in there, but you don't have to pay attention to that. That's just minor detail. So TGF beta, for example, as I said, that is it's a negative regulator. So if you look at the TGF beta, when it binds to the receptor, it enhances the, it blocks the cyclin D-CDK binding, which is going to inhibit cell cycle, inhibit the cell proliferation. So this is all growth factors, EGF, the PDGF, all others we talked about. They are all mitogenic. TGF beta is suppressor. TGF beta is not a mitogen, it's an inhibitor, and TGF beta uses a different system. Okay. Now we talked about the receptor by itself. Now what is signal transduction? Signal transduction is a process by which cell converts an extracellular signal into response. Our final product is the response, right? So that is the response, meaning it's a cell-to-cell communication, which is how it responds to the environment, what happens to the internal processes. That all requires a signal transduction. It's all called signal transduction. When you look at a single layout, you have a signal, you have a receptor, you have a signaling cascade, and then your targets will become like metabolic enzymes, gene regulator, or cytoskeleton proteins, and the final product will be either altered metabolism, altered gene expression, or altered cell structural differences like the laminin and fibronectin and things like that will be changed here. So those are the responses that we're talking about. Okay, there are many proteins that are involved in the process once the receptor is bound to growth factor, and after it's phosphorylated. What happens is that there are many. I can read this. That's why I'm looking in my things, right? Because the color. So you adapter proteins. Those are called adapter proteins right here. GRUBs, GRB2. This one is the adapter protein. And then what happens is that it has to go through the process. Many of the protein kinases involved in the process. It increases regulates the MAP kinases. The MAP kinases get, if you look at that, several MAP kinases. So this is called MAP KK. Before that there is MAP KKK, like 3 Ks, and it gets into the MAP KK by phosphorylations. They become like kinases get phosphorylated. Then again it becomes gets phosphorylated, become MAP kinase, and they get into the transcription factors and into the nucleus. So how does that happen? This is a really important slide. The functioning of a RAS, for example, here we are talking about RAS. So when RAS is complexed with a GDP, it's inactive, and when the RAS complexed with GTP, it is active. So the RAS functioning requires the engagement with GTP, which allows the phosphorylation of RAS for the downstream signaling. Now this happens by there is a thing called GEF, or guanine nucleotide exchange factor, right? And GAP, which will work here, is called the GTP, is activating protein. So this is a cycle which continuously goes on. So you have a RAS, inactive RAS, you have a stimulatory signal from GEF, from GDP comes up, GTP gets involved, this gets phosphorylated, then phosphorylated one, it goes up and makes RAS inactive, does the functions in between, GTP gets hydrolyzed, and by using the GAP. Now this one is the RAS activation on and off function. You have a trigger that turns on RAS, or that is a trigger that turns off RAS. And turning on and off is very important for the functioning of a RAS. So and then all oncogenes are going to work in this fashion. They may require different systems, but the RAS requires GAP, which has a lot of literature on it. Okay, so how can you determine the growth factor function? You have your control where you have a growth factor and you have a phosphorylated catalytic active unit. You remember, if cascade, you can generate a knockout mutant. So now you have a mutant that's a knockout, so the growth factor activity is not occurring. So you look at the animal that may have, or cell that does not have a growth factor effect in that growth factor receptor knockout. In that, you look at the activity of your compound, whether it still works or it doesn't work, but you can identify the growth factor activity, or you can overexpress growth factor by some mechanism. The overexpression, we do overexpression by many different ways. You can take a cell where you identified overexpression and you clone them, or you can have a compound that helps in overexpressing the growth factor response. So you enhance the growth factor overexpression, and you can study whether if you overexpress growth factor in the cell culture, whether your response changes. You can block growth factor receptor by using antibodies. So now your growth factor effect is blocked because the signaling is not going to go through. So that's how you study the function. And also you can block the growth factor by a molecule that blocks the growth factor, and that's a therapeutic approach where you identify a compound or a drug that's going to mask the growth factor, and it can prevent its signaling, so it does not do its job, right? So that all happens at the cell membrane level. The procedures that you need to study are, Western blot analysis, which everybody does, immunochemistry, immunofluorescence, using the fluorescing antibodies or the regular antibodies. You use the siRNA, you look at the gene expression, and you look at the real-time PCRs. Those are the procedures that you need to use. I don't know whether in the labs here I'm sure that you guys use the Western blot analysis. I don't know whether you use RT-PCRs or the qRT-PCR, real-time PCRs and things like that. Those are important for many studies. Okay, this is one of the examples for colon cancer studies. For the things that are colon cancer, this is beta-catenin is very important. So there is a protein called Wnt, and they have Wnt genes, and it's a protein. So people measure the Wnt gene mostly, and there are 7-8 different isoforms of Wnt's. So in the absence of Wnt, there is called a frizzled protein here, and there is another protein called the Dishweld. Now this complex here, the axine, APC, this GSK3 beta, and that is phosphorylated GSK3 beta and beta-catenin, they bind, right? So when the complex is formed, and that triggers the degradation of beta-catenin. Okay, beta-catenin gets degraded. Now if you have a Wnt binds to frizzled protein, that phosphorylates frizzled, it in terms activates the dish vessel, gets also phosphorylated, that makes this GSK3 beta inactive, so its phosphorylation ability is gone. Now you have a beta-catenin that cannot bind, that cannot work with the complex. So beta-catenin comes off, and that promotes cell proliferation. So in colon cancer studies, it's very important, and if we have time on the last day when I'll talk about how everything gets together. I've brought in several different experiments together from my lab to show how everything works, and all these things that we have done in the lab, like we have done the Wnt and growth factors and siRNAs and just name it. So as the time comes, what I'll show you is that everything that we are learning, a practical example. I'll go through about hour and a half of couple of different compounds to see how all this comes together, all this fits together. Okay, and this slide shows the functional significance of various styles of receptors here, all the EGF receptor and the bunch of viral component opposites of that one, but not in viruses. These are like derived from there or like that, and these are the ligands. This is what you see, the overexpressions, and these are the types of tumors you get. And this is again, I got it from the Weinberg book. If you get hold of Weinberg book, it's like a Bible of cancer biology book. It's the best written book. And then if you look at the signaling pathways, there are 3 different signaling pathways that we put up here, and all 3. The reason I put these 3 is not for any other reason, but people have used it a lot. So PI3K process, all 3 are regulated by RAS, but PI3K pathway goes through the AKT and PKB. PKB is like a protein kinase B, and then it goes through the some sort of growth factors here, and it does the GSK3 beta stimulation. So cell proliferation, cell growth, the mTOR pathways, and that's apoptotic inhibitor, which inhibits apoptosis. So what happens is, as I was telling you just a while back, that you identify something that your compound does. So I had a PhD student who finished, Akash. So he was looking at one of the compounds called ProLX. It's a therapeutic compound, and looking at ProLX and its effects on aromatase inhibition. And what he found while doing it is that it was changing the mTOR signaling. So he spent he had a habit of doing many things which was not good, but he spent like 6 months doing mTOR signaling. Well, results came out good, but what I'm trying to say is that you look at something, what it does. You have cell line, you grow with this, and say, he asked me, he says, can I do the mTOR signaling? I said, you can try it out, see if it works. Well, at the end, that didn't pan out. I mean, he was working on mTOR, and then he had to switch gear and start working on totally different signaling pathways. Now he's doing postdoc in Arizona. So that happens in many different ways. Okay. Now this is the last topic within the growth factors that we're talking about. So if you look at that in growth factors, there are some, not even in growth factor, in general, okay? We all know about gene expressions or gene regulation, that you have a compound or you have a process where gene is expressed, and then you have overexpression of genes, underexpression of genes, and the gene can do the protein, and then you have a different response. There are immediate early genes, which are the genes which are the final gene product, or they're called delayed genes, are regulated by early genes. So this is very important for your studies if you guys are doing. So like if you look at something within the first 10, 15, 18 hours of your studies, not even that, even earlier than that, within like few hours after you start your cell culture, the genes that are expressed, or you see overexpression of genes, those are not the genes you're going to find a day later. Okay? Those genes are not going to show up. So those are called the immediate early genes. And and I did not know about this when we had, in our lab, and then somebody had showed that, oh, okay, did you know this? And I said, well, this is really good, because they'll come and show the data that, okay, if you do it for first 15-30 minutes, you have burst of genes that are coming up, and they're going away, and the new genes come up. So we thought that, well, there's some errors in the experiments, why it's not coming, we can see, but then it's lost, what's happening. And these are called the immediate early genes, and I'll show you what happens in them. These are some of the immediate early genes. I'm not going to go through the list of it, but then here is the process. So when you have the so the delayed genes are like really dependent on the early genes. So you have a growth factor receptor is firing, you have the early gene synthesis once it goes through the cascade, signaling cascade, and you have the activated transcription factors, you have immediate early genes. Now those messenger RNAs of immediate early genes are again going to make proteins, and those proteins are going to serve as transcription factors for the subsequent genes, and those are called delayed genes. So like when we are doing cultures or these studies, after 3 days you incubate, which is normal practice. Okay, you are looking at after 3 days or after 2 days of your incubation to look at the gene expression. It may be the original gene, which may not require early gene in order for it to function or to come up, but it's also likely that that may be a function, or there is an earlier regulation before that, which is regulated by a new protein, which is not your original protein, but that protein is coming from the immediate early genes. And this is a road map. I like this slide. We're not going to go through that, but this one is a good subway map, he says. So it's like a train system. So you have to, you go to your endpoint by, you just follow it up and go through, change the train here and go there. So these are all regulatory. That keeps on getting changed every time. Like people put in new in database and they come up with a new system. So this is a wind pathway, for example, we just talked about. This is a TGF beta pathway and it goes through the system. These are some other growth factors. These are cytokines. So if you look at the, if you follow it through, you will get a good picture. But there is no sense in my reading off, because it's all it is a signaling of various molecules and various genes in the system. Okay. So now we're going to turn our attention and go from growth factor to cell apoptosis. When are we? I can keep on going, right? What's the stop time? Anybody? Up to 1? Okay, good. So we can do apoptosis now. Okay. Now cell apoptosis, which is I did this slide before, but I will review it. But cell apoptosis, what we're going to talk about is the programmed cell death, and it is also a very complex subject. And if I do that, it becomes very boring, because they have the PARP cleavage, and PARP cleavage is so complex. So I won't go into that. I have slides. If somebody is interested, I can explain in detail, but I'll go over very very briefly over there. Okay. This we talked about in last class. So the key characteristics differences between normal and cancer cell is contact cell inhibition, which is really signature for cancer cell. Normal cells know their limit when the cells grow. When it comes near the other cell, it stops piling over. There is a contact inhibition, whereas the cancer cell has a loss of contact inhibition. Okay. Growth factor requirements, normal cells require it, cancer cells don't require it, because that have as we talked about before, it will come back to you that it is self-propagating. Anchorage dependence is present and anchorage dependent is absent. So what that means is that the normal cells cannot pile up on itself, but the cancer cells can pile up, and that's how it can form tumor. Then you have the cell cycle checkpoints. They're not really absent. It's that's a misnomer here, but all it is it's abnormal. So cell cycle checkpoint controls are sort of lost, and karyotypic profile is normal or karyotypic profile is abnormal, which is also not true. It could be, but it doesn't have to be. The karyotypic profiling is still could have a 23 pairs of chromosomes and everything else, but and the chromosomes can look normal. They don't look like broken up or damaged or anything like that. So we won't call it abnormal. Now you can call it abnormal from the point of view that it has a different function. The function is lost, so it's abnormal. But structurally, they still are, your 23 pairs. And proliferative life, this is important. We discussed about it yesterday, that proliferative lifespan of normal is finite, and cancer cell is indefinite. Now, yesterday we were discussing, about the normal cell, that CHO cells we were talking about, and then I was thinking about that also later on in my mind. Something sticks in, then it doesn't go away, right? So I remember that in literature, and you can check it carefully. Many people have referred to normal-looking cells that are immortalized as near normal. There's a normal like. So if you write a grant application, make sure that you don't call it normal, because I remember I'm on the review committee of many NCI committees, National Cancer Institute, and also in USDA and things like that. So in one of the times I remember that there was a colon normal cell. Somebody had made a big thing about it, and then there was a huge big discussion about whether you can call it normal, and it had come up that you can call it normal like. So that's the best word to use. You say normal / normal like. So that means you know that this is not normal, but it has it has maintained majority of the normal functions. It's not normal because it's because it's growing indefinitely. It cannot be normal. Okay, there's another. I saw something here. It says normal cells failed to undergo apoptosis. Not true. I've seen cancer cells getting apoptotic signals. In other words, I can see program cells. If you look at if you look on the PCR and you look at the band profile, when you have a apoptosis occurring in the cell, and you take a DNA and you load on your PCR, you will see these exactly known size fragments. You can see the bands, 4 or 5 bands, and otherwise you can see the lump either in the bottom or you can see the smear. So, so if you look at a cancer cell and you can see apoptosis, and I give you an example. Say you have a compound, right, and then you want to taste it. Well, what cell line you test on? You test on MCF7 breast cancer cell lines, on HL60, on cancer cells, right? Well, something that you are testing your cancer preventive agents on a cancer cell line, that is not preventive, is it? It's really therapeutic, because you're working on a cancer cell. You're not working on a normal cell. And then you look at a cancer cell, you treat with the drug, which you call it a cancer, and cancer prevention is close to my heart. I spent a lot of years, but I also wrote a review on this. So I can take a claim. I said exactly the same thing, that cancer prevention, and I'll show you tomorrow or day after tomorrow, that cancer prevention, the reason I had written this is because I wanted to make a case that I wanted to use a drug compound that we confirmed as a cancer preventive compound. I wanted to write a grant and call it a cancer therapeutic, because I wanted to make it therapeutic into it. So I started writing saying that, well, since all the effects of cancer preventive agents that we looked at were looked on cancer cells, which means it's a therapeutic agent because it inhibits the cancer cell growth, and something that inhibits a cancer cell growth is cancer therapeutic. So after qualifying that, if you start writing, now it's just like when we were little, you prepare an essay on a cow, and the questions come on a farm. Well, okay, you have a cow that goes in the farm and eats the grass. You can change it to a cow or a grass, right? And write the whole essay, and you still can get away with it. Same thing happens here. So in a logical way, I'm talking about logical way. So from an apoptosis point of view, most people, 95% of the literature that you will see, they have a cancer cell, they put a compound on it, therapeutic compound, they look at cell apoptosis, and they find apoptosis with variety of different methods. That means cancer cells apoptotize, right? That means there is apoptosis in cancer cells. So this is wrong. Okay, I took it from somebody's thing. This one here. So it doesn't fail to undergo apoptosis. Okay. So here we just are going to say that, well, the cells, normal cells, they live for a given period of time and then they die. That means the normal cells don't grow past few generations, they all die off. They have a signal to die. And that thus through apoptosis, you have a cell apoptosis occurring, which means a programmed cell death. Now, we won't have time because it's only, we just have very limited time. So, which I spend a whole hour in my class is telomerases. So the telomers are the, they're the ending, they're telomers at the end of the chromosomal thing. So what happens is, it's an aging thing. When the telomers start coming off, and as you age, the telomers go away. Now in the cancers, the telomere process is sometimes stopped, and your telomeres don't become shorter, and it remains cancerous. So that's another process, another thing that goes on in the system. And so what is apoptosis is a physiological cell death, and it's called programmed cell death also. It's a cell suicide, cell deletion, or a variety of different words that are used. How does the apoptosis how do they come about? The major reasons of apoptosis to occur is the aging, of course.
Anti-aging, they have tried to prevent this telomere shortening. So does that mean that they are promoting cancer, or what?
No, the not having telomere, that means the cell's survival signal is maintained, right? Now, don't really have not having telomeres or
Preventing telomere shortening.
that's what I'm saying. So just to the response that have the telomere shortening is that's going to decrease your aging, but that's a normal process. So what we are saying is the telomerase shortening is not really the cause for tumor, right? That's a event. So that happens when the cancer happens. It's like correlation. But in case of aging, that's altogether different. It's just like two different things we are comparing. But the correlation is there, but it's like it's a little bit far-reached in a little bit. Okay. So the apoptosis can occur by a number of different ways. You can remove the growth factors. This is very simple. So what happens is that you can have the stress factors. You can have something that's like dieting, something that reduces somehow the normal cell deletion or growth or suppression. That's all going to cause programmed cell death. This occurs in the embryonic and adult cells, and proteins involved are always present in cell that needs to be activated. That's obvious, because you're going to see apoptosis where you're going to show the inactivation. So you're removing the factor that activates the cell. Then you just remove it and you have the suppression, right? Now there is another word, and we know that necrosis occurs. In cancer cells, you always have a necrosis occurring. And you can see, like when you look at the center of a tumor, you have a cell necrosis occurring, and we'll be talking about it. That also has to do with hypoxia, and I'll be talking about that a little bit later. But the comparison, if you look at apoptosis and necrosis, the comparison, so the apoptosis is an actively regulated process. Cell death is due to injury or lack of oxygen or lack of some sort of a some factors, and I'll be talking about it, like HIFs and things like that. So they have the unregulated cell death to injury. You have DNA fragmentation occurring. So as I was telling you that when you do the PCR of DNA, you will see the bands of certain size on your PCR, Whereas in case of cell necrosis, you will have the smear. It will have a big smear coming here. So you have the fragmentation of nuclei or cell shrinks. So like if you look at the cells undergoing apoptosis under the microscope, you will see the shrinkage. You can also see, and I'll show you some pictures of apoptosis. So if you stain the cells with acridine orange, and then you look at the cells under the scope, you will see different colors. So the apoptotic cells will show a different color as compared to the dying cell. Then you have the apoptotic bodies. Now apoptotic bodies, I'll talk a little bit but not much, because that you have the path cleavage occurring here, and the phagocytic nature, that's not within the scope of this class. Okay. And this is the same type of thing, which is all, you can look up. I have this slide in your brochure, and there's a comparison between the two. The major ones that we already talked about in programmed cell death, the morphological changes that we see in terms of apoptosis or necrosis is cell volume is decreased. In necrosis, the cells swell. The condensed chromatin is another feature that you see in apoptosis, whereas the chromatin is fragmented here. The mitochondria is you have a really known structure of mitochondrial structure that is normal here before it starts breaking down, whereas here is aberrant. And you can go through these. These are all there are certain genes, there are specific genes required for apoptosis. There's no certain genes required for necrosis. And the ion pump means the channel pumps are lost. Okay, this is some of the pictures, but this is not a really good picture, so I'm going to talk. Okay, so this is the experiment that that was done, and it's in the literature. So you have the eggs, right? With the early cyst, these eggs are injected with the early stage blastocysts, have we talked about in the first class. And so what happens is that if you look at the genes, the embryos that carry the transgene from this blastocystic thing, they breed with each other. Some will cause cancer, like for example the transgenes. So if you look at from the beginning, you have a clone genes that are injected in here, which are in case of all MEK and BCL. I was just trying to see what genes they used. MEK and BCL, I didn't remember this slide for too long now. So you have a genes that are involved, injected. You have single genes, either MEK injected or BCL2 injected in this mouse, the cells containing these genes. And then you go over, you have the carrying only one gene here, and here you have the breed them, you have both genes expressing in this one. Now you look at the tumor promoting profile or cell, or the mice surviving, but the one with BCL2 alone, they survive longer, and they die, and MEK with BCL2 they die. Okay, so what does all this mean? What we're trying to say is BCL2 is a survival gene, and it's used in most of the studies that we do. You use the genes or the proteins that are involved with bad BCL2. BCL2 is a survival gene. So when you put the animal, when you add the gene or transfect the animals with the cells with the survival gene, and then you transplant those cells into the animals that are carrying the survival gene, the genes have a tendency to live longer. I mean, the animals live longer. But if you put the oncogene in there, which is going to make the lifespan shorter, they are going to die. And when they have the two combined, the MEK is expressing more of its action than BCL2, and you have an intermediate result. That's all they're trying to show. Okay, so there are types of apoptotic genes. There are pro-survival genes and there are pro-apoptotic genes. So you have BAX and BH3 family. BAX is very popular and BCL2 is very popular and BAD is very popular. So if you are going to look at the studies, we are going to measure some sort of expression of apoptotic genes or apoptotic proteins. You're going to select BCL2 expression, you're going to select BAX expression, and you're going to select BAD expression. And between the expressions of these three, you can say, how proficiently the cells are undergoing apoptosis, or how the cells are going to survive. Right? This is a layout, so we don't want to go into that. Okay. So some of the slides here are things that I did not have in here. We'll skip this. Okay. So now if you look at the summary of this, then you have pro-apoptotic genes, which are involved in terms of preventing apoptosis. Then you have a pro-survival genes, and this one is really used a lot in the literature now, is BCL. And what they do is the function of the pro-survival gene is to suppress the BAX expression, and BAX is a pro-apoptotic genes, right? And then the BAX. So for example, when the survival gene is enhancing the expression of BAX, because it's going to prevent the survival function of it, you're going to get the apoptosis. And then you have the pro-apoptotic genes which work on the BCL2 itself. This is one of the examples that this is from my lab. The work on dagulin. So this is a MDA-MB231 cells. Those cells are called triple negative breast cancer cells. And if you look at the effect of dagulin, this is a control, and this is a treatment with dagulin. So you have the apoptotic process restored. Now the apoptotic, so that this kind of gives the, I did not remember whether I had this slide in here, but what this shows is that the cancer cells, if you look at the normal cell, for example, and do a control profiling like this, you won't see apoptosis like that. There's no apoptosis, or cancer cells don't have apoptosis like that. But the cancer cells do undergo apoptosis, is shown by here. So when the apoptosis occurs, there are really known fragments. Otherwise, the control has everything on the top because it just does not migrate. But if you have something that does not induce apoptosis, you will see a smear of DNA. You won't see a selective bands. So that's the process. That's a very easy process, and you use if you're going to show that you want to study apoptosis. Oh, I thought that I did not have this slide either. Okay. So I wanted to show in this one is, remember I told you just 2-3 minutes ago that I'll show you a couple of days after, but I did not go through these slides before, because I didn't think that I was going to talk about apoptosis today. So when you have a control, and if you stain the cells with acridine orange, okay, then you're going to see the control cells are going to look normal cells, growing cells are going to be showing green. You're going to have the yellow cells which are undergoing cell apoptosis, and the red cells which are dead cells. Those are the cells that are dying. And this is this work was done by one of my MD PhD student, Julie Choi. We published this some time back, and she was working on a compound called ixocarpalactone. And ixocarpalactone is found in salsa. in when we have this chips and salsa. This salsa, green salsa is made from fruit called tomatillo, not our tomatoes, but tomatillos. And that tomatillos, when somebody had done the long, my friend Kinghorn had done that. So done the long, some guy did a PhD and said, well, ixocarpalactone looks good. So the ixocarpalactone, Julie studied for some time when she was undergraduate in my class, in my lab, and she looked at the effect of ixocarpalactone on the cell apoptosis, and you can see that when ixocarpalactone was used at 5 micromolar, she was getting this effect, and in the same effect. This the 5 is the same thing, different pictures. Control cells, and the one with the ixocarpalactone. So, and she got a paper out of this also. Okay. So I think that I'm going to stop because our apoptosis thing is done. Because if I get into the P53, it's going to be another long time. If you have any questions, I can answer. So you can do that for 5 minutes.
So what is necroptosis? Is a new term, necroptosis.
So I think it's a fancy term. By causing the tumor cells are going to undergo necrosis, right? And the necrotic cells also may have a combination. The apoptotic cells sometimes just kind of have nuclear shrink and go away. Sometimes they do become necrotic by having the lack of the oxygen, the hypoxic condition. You have HIF, not having any activity in there, and sort of a combination of apoptotic necrosis, I believe. I really have not come across a problem, but that would be my best guess.
Answer.
Okay, so what we're going to do in the afternoon is following. We're going to show you short clips of normal cancer cells. I have a short, this short is real short, couple of minutes videos in there. I have one on epidemiology, and then we have one on cell regulation, the one I talked about, and one on signaling. So we'll look about 10-15 minutes of video, 10 minutes, and then we discuss that to see, because this video is also not, that's the one that's not I generated. I'm just going to, we're going to show, and so I'm new to them as much as we are, so we'll discuss that what is in there that may be happening or not happening. And so that will be our afternoon session.